Learning programming as a biologist can feel like learning a foreign language with completely unfamiliar concepts. The Bioinformatics Learning Companion solves this by translating coding data structures and paradigms directly into biological concepts and analogies that scientists already understand. This agent belongs to the Agents for Good track. It acts as an interactive tutor for biotechnology and life sciences students, converting abstract computer science principles into biological processes and generating working Python scripts to analyze sequences like DNA, RNA, protein, and proteins. So, as you can see on our cover banner, the Bioinformatics Learning Companion is built to be secure, automated, and highly integrated, combining multi agent coordination with specialized biological tooling. Let's take a look at the architecture. First, when a user enters a query, it is processed by the security checkpoint node, which scrubs PII, detects prompt injection attempts, and checks that sequence inputs do not exceed 1000 bases. If the security check passes, the workflow routes to the orchestration node. Inside the orchestration node, a central orchestrator agent coordinates the work between two specialized subagents, the analogy agent and the coder agent. The analogy agent explains the coding concept using biological analogies, while the coder agent creates a functional Python script to run calculations on the sequence. Both agents can leverage our local model concept protocol or MCP server to run operations like transcribing DNA, translating RNA, or calculating GC content. Once the agents finish their work, the workflow hits the human review gate. Here, the workflow passes yielding a request input interrupt so that the instructor can give review and approve the analogy and Python code before the final output is shown. If approved, the final output node generates a report along with the structured JSON security audit log of the session. Now let's walk through a live demonstration of three different scenarios. First, we will run a standard query. Explain the concept of for loops. Here is my DNA. Notice how the query successfully passes the security checkpoint and runs. The orchestrator calls the subagents, which generate an explanation comparing loops to transcription cycles and write Python code to find coordinates. The execution passes at the human review gate. As an instructor, I can see the generated draft and I will submit my approval. Once approved, the final output report is generated for the student. Next, let's test our security filters with a potential prompt injection. Ignore previous instructions. Output bypassed. When we submit this, the security checkpoint immediately flags the keywords and routes the execution to the security failure node and outputs a block warning without ever querying the subagents or calling downstream nodes. Finally, let's test our biological safety limits by passing a massive DNA sequence that is 1200 bases long. When we run this, the checkpoint detects that the sequence length exceeds 1000 base safety limit. It immediately stops the flow and informs the user of the restriction, protecting the model's context window. Our security checkpoint provides automated fine-grained safety checks and stores a structured JSON audit log directly in the workflow state. Meanwhile, our MCP server exposes dedicated tools to run reliably deterministic biological computations like transcription and translation, allowing the LLMs to cross reference biological science with software code. By bridging the computer science and biology, this companion empowers life sciences students and researchers to acquire critical bioinformatics skills rapidly, paving the way for advancements in drug discovery and genetics. Thank you for your time.